Our honorable speakers, colleagues, participants, ladies and gentlemen, a very good day to everyone. I am Nihan Sultan. I will be host for the day. I would like to announce housekeeping messages regarding our symposium. To ensure a smooth running of the workshop, please make sure you have access to a good Wi-Fi or cable internet connection. We kindly request that you change your Zoom profile name to your full name to ease name tracking for attendance purpose. The participant certificates will be only issued upon attaining all 3 days attendance. The sessions will be our live streaming on YouTube. For those who cannot get into the Zoom due to any technical issues, please join there and inform us. Please join our Discord server for announcements and other communication. If you have any technical issues, kindly seek help through the chat box or email to us. If you would like to ask questions to our speakers, please type them in the chat. We have 3 keynote talks today. Our first event for this morning is a keynote talk by Dr. Denis Bauer. Then we have our Ezgi Turkey presentation by Sevillay Gulesen. After that, we will have a panel session on current topics on computational biology with Dr. Beste Turanli, Dr. Ezgi Karaca, and Dr. Tunca Dogan. Then we will have a keynote talk by Yuval Itan. Finally, we will have an award ceremony for Best 3 poster and oral presentation. Now, I would like to welcome our chairman for this morning keynote talk session, Dr. Osman Akçakır. I hand over the platform to Dr. Osman, who will continue from here. Over to you, Dr. Osman.
Thank you. Good morning, good afternoon, everybody. I'd like to introduce our keynote speaker, Dr. Denis Bauer. Dr. Denis Bauer is an internationally recognized expert in machine learning applied to processing big genomic data. Her achievements include developing an open source, artificial intelligence-based search engine that helps researchers pinpoint the exact genes they need to study or edit to cure disease. She is a transformational bioinformatics leader at CSIRO, Australia, and adjunct associate professor at Macquarie University, also in Australia. At CSIRO, she is involved in Australian and international initiatives to integrate genomics into medical practice. Her achievements have been featured in international press such as Genome Web, ZDNet, Computer World, CIO Magazine, the AWS Jeff Barr blog, and was in Computer Weekly's Top 10 IT Stories of 2017. With that, I'd like to invite Dr. Bauer for her speech.
Thank you very much for the warm welcome. It's fantastic to be here with you today, if only virtual. So with that, let me start sharing my screen and then we can get underway. Good. So today's topic, linking in with the rest of the symposium, is back to the fundamentals. So science is transforming. I love the interaction here. Science is transforming in that we are required to produce results faster, and there's ever more data than ever before, which in my mind really means that we need to go back to the fundamentals of producing better algorithms, looking at scalable data structures, and most of all, collaborate more collegially internationally. So therefore, with that, I want to tell you 3 stories. The first one is around efficient algorithms and how we can identify disease genes more effectively. The next one is looking at scalable data structures, and specifically here for genome engineering, but also COVID, looking at the COVID data that scales or is increasing at an ever faster rate. And then lastly, looking at collaborations and how we can do them differently using novel technologies, specifically cloud computing. But before we go there, allow me to introduce the research organization that I'm part of, which is CSIRO, which is Australia's government research agency. At CSIRO, we're really passionate about translating research into products that people can use in their everyday lives. One of the most famous products, of course, is Wi-Fi, which CSIRO invented together with Macquarie University, which is now used in more than 5 billion devices worldwide. More on a health note, the eHealth Research Center, which I'm part of, developed the first clinically validated mobile health app for heart rehabilitation, CardiHab. And then on a lighter note, we developed together with Samri the Total Well-Being Diet, whose recipe books are now on the book bestseller list alongside books like Harry Potter and the Da Vinci Code. So I think from that perspective, we do have a nice balance between products that people need and products that people enjoy. Now, let me start by a statement in that AI is a new electricity. And one of the founders here in Learning AI and Deep Learning, he was saying that I can hardly imagine an industry where AI is not a transformative part of. And I think health and research is going to be one of those areas where AI is really making a difference. But let's put that as a poll out to a question to you. So I figured it would be good to make this a little bit more interactive. So you should see a poll pop up where you can let me know whether you think machine learning and AI is really transformative or whether it's just a buzzword. So I'll give you a little bit of time of answering those. Remember, there's no right and wrong answer. It's just a little bit of fun along the side. Therefore, I will be going quite quickly through those quizzes because it's just a check-in along the way. So it looks like the majority of you say that machine learning or AI is going to be transformative. Well, I tend to agree with you. And especially, I think it will be transformative in genomic research. Now, the reason for that, I think, is that genomic research has more data than ever before, and it has problems that are really amenable to machine learning. Specifically, the cohort sizes are getting larger, and we want to look at gene-gene interactions, so really complex interactions in the genome. Sorry, problems here, going to the next slide. All right, so, and the reason that machine learning is going to be better done nowadays than I think it was in the past is because we have these new ways of computing data, massive amounts of data. And specifically here, I'm talking about Apache Spark. And allow me to walk you through how I think about Apache Spark by making an analogy to high-performance compute. So probably the majority of people in the audience will be familiar with high-performance compute, where you have these large clusters of really bespoke and specialized CPUs that have multiple CPUs on an individual node. And you string a lot of those together in high-performance compute facilities in order to achieve the compute-intensive grunt that you need for traditional compute-intensive tasks. The problem with that, though, is that in genomics, it's not a compute-intensive task, it's a data-intensive task. And therefore, we tend to have problems fitting all of that data into a single node, into the memory of a single node, and having the CPUs associated with a node, which means we typically run into the problem of stringing multiple nodes of those together. And if we do that, we do have to implement a bespoke way of translating or brokering the data exchange between the nodes. And this is exactly where Apache Spark comes in. In my mind, it's basically dissolving the boundaries between nodes and uses the commodity hardware and their CPUs in a native standardized way, so we can use all the CPUs in a distributed cluster more seamlessly. And to me, this is more amenable to data-intensive tasks like genomics. So here we developed Variant Spark, which is a machine learning method using these distributed ideas behind it. And this is why I bring this up in terms of better algorithms. So we're using Variant Spark, which is basically a random forest, but we implemented it differently. Typically, you scale out random forests by scaling up or by doing the parallelization over the nodes or by distributing or partitioning the data horizontally. But again, in genomics, this horizontal partitioning is not really fitting into the node structure. And even with distributed computing like Spark, that's a problem. So what we've done is we partitioned the data vertically, and therefore we can use more of these nodes in a distributed way in order to calculate in a parallelized way those random forest trees. And what we showed is that with traditional dataset sizes, we are 3.6 times faster, but the key is that it can scale up to future dataset sizes of 1 trillion data points, and it can compute that in 15 hours rather than 100,000 years, what the other tools would use. And again, coming back to different algorithms to cater for different problems, like these newer problems that have different properties. Also, we're using the cloud in order to do this. Specifically, we're using a MapReduce cluster, which is a managed Spark instance in the cloud, which is shown here in the middle. And then that can link up to individual buckets where the data is located in the cloud. And this is then natively scalable to large data volumes that future studies might have. But coming back to the task at hand, what is Variant Spark doing and how does it find disease genes? So traditionally, when we find disease genes, we have each line here is a genome, and then each box represents a mutation in the genome of individual people, of cases and controls. And then in the Mendelian case, we're looking at each location independently and identifying the ones that are enriched, where the mutations are enriched in cases versus controls. But with a lot of complex diseases that are multigenic, where individual locations in the genome contribute to it, and not in an additive way, but in a more complex way, this individual looking at this breaks down. So specifically, you might have gene A, B, and C, or SNP A, B, and C, and then together they have a certain additive effect. But in specific diseases, there might be a complex interactions between them, which have even a higher association power. So even though A, B, and C independently or A, B, and C additively might not drive the disease, the complex interaction between them might. And therefore, in the traditional way, we would have kicked them out because they never reached genome-wide significance. Whereas looking at the genome with a machine learning approach that takes all those locations simultaneously into account, we can find those complex interactions that only together drive the disease, but individually have no effect. Therefore, applying this new way of thinking to genomic data, we first applied it to motor neuron disease. And here we looked at 15,000 case controls in order to identify the disease genes and the locations that together drive the disease. Now, we found some new associations which together with other regions in the genome might drive the disease, and that obviously needs to be confirmed with more biological results in the future. But it shows that there is a new way of really looking at this, at the genome together and simultaneously in order to find these small effect sizes that together might drive the disease. Now, because everything is scalable and we have this new algorithm structure that partitions the data vertically, we're able to scale this up to now 50,000 case control studies. So this is to get together with the UK Biobank and looking at heart disease. So 50,000 case control individual using Variant Spark, and we can then plot basically in a traditional Manhattan GWAS style plot the locations in the genome that drive the disease. And then from there, we can use BitEpi, which takes these locations that were flagged to have some contribution in order to identify which ones together drive the disease and come up with sort of a network approach from there. And what we can see in here is that we identified certain areas that are already known to drive cardiovascular disease, but we also have locations that interact with those and therefore amplify either by exacerbating or by forming resilience factors in order to modulate the severity of the disease. Now, this is obviously not only applicable to human genomic data. So what we've done now is to look at the COVID genome, the COVID virus genome. And here we have 5 million samples from all around the world as part of the GIS8 consortium. So what we wanted to do is we wanted to identify the individuals who have severe COVID disease and look at the genome of those patients, the virus of those patients that have infected those patients, and compare them to individuals where the disease was mild. Now remember, we had 5,000 samples to choose from, but in the end, we could only use 5,000 in order to identify or to do this study, which means the majority of samples that are in GIS8 are not annotated sufficiently for us to say this is a severe case and this is a mild case. But 5,000 is not to be sneezed at. 5,000 is a good, a moderately good sample size, and we do have some preliminary data. Now, launching the next one. Again, remember, this is a little bit of fun on the side. Which region of the genome do you think is most important? No, sorry, my apologies. I seem to have skipped over some of them. Ignore this one that is up here, and maybe I can ask one of the moderators to fix this issue. So just in your head, which region do you think is most important? And allow me to go to the next slide and tell you that, of course, the spike region is one of the areas that has come up, which is not surprisingly, the spike region is the one that helps the virus get into the cell and attach and recognize the right cells. But interestingly, there are some other associations with the membrane protein and some other locations in the virus genome that seem to have some influence of the disease severity for humans. Good. So this brings me to the second story, which is around data structures, scalable data structures. And the example here that I want to bring is for CRISPR. And Jennifer Doudna was saying that the world around us is being revolutionized by CRISPR, whether we're ready or not. And here's the next quiz. Let me see if that, yeah. I don't think that's actually all the quizzes that I put forward are actually in here. So again, in your head. Oh, fantastic. Yes. Are you ready for genome engineering? So seeing if you can click on the poll already. I might let that poll run in the background, see if it's running or not. And in your head, again, answer this question, because again, there is no right and wrong answer. Now, where we are using CRISPR genome engineering for is for treating childhood diseases, specifically a specific liver disease. So this is together with the Children's Medical Research Institute here in Sydney, and there is a condition where a single mutation in the genome causes a severe liver disease that is currently not treatable. Well, the treatment for it is a liver transplant. So in newborn babies, having a liver transplant is obviously not the ideal outcome or not even a good treatment. Therefore, having an alternative, which is here done over CRISPR engineering, where the molecular machinery CRISPR is put into into an envelope, and that envelope is only delivered to liver cells. And in there is only going to that specific location where the mutation, the misspelling is located in order to correct that. Now, obviously, this is still in clinical trial. And as you probably know, for the ones that are familiar with clinical trials, it's a long way until this will be in clinical practice, because there's a lot of things that need to be optimized along the way. A lot of those things can be optimized from a bioinformatics perspective, and this is where we are interested in helping out as well. So obviously the first task for things like that are to identify the exact gene that is involved in the disease. Then we need to identify how to target this particular gene, like which CRISPR binding site is the most efficient one in order to target that. Then we need to develop the best capsid in order to deliver that very accurate CRISPR mechanism to the tissue that is affected. From there, the question is, can we generalize so that it's working for or the patients that we want to be treating, rather than the individual that we have the reference genome for example. And then, of course, it's around the safety of the delivery vector. For example, if we're using AAV, that is a virus, do those virus elements integrate into the genome of the patient and cause, for example, cancer later on? So is the cure worse than the actual disease? Now, focusing on finding the optimal target site in the genome, the way I think about it is sort of like a sand on the beach. You want to find the right grain of sand on the beach that has the right shape, size, and color for the genome engineering mechanism to interact with it. But also it needs to be unique enough so that it doesn't go accidentally to other locations, like off targets, and accidentally destroys other genes. And obviously there's a whole variety of different scenarios. And you can look at the individual tissues, you can look at the individual engineering mechanisms, or prime editing, Cas9 editing, and so on. So for all of those, we developed individual machine learning methods that were trained on the data set specific to that scenario, and then can only be applied to other cases of that scenario. So therefore, we developed a whole suite of tools in the GT-Scan app suite that can do that. Now, remember, this section is about efficient and scalable data structures. So what we were doing here is using serverless applications in order to do that. And we were the first to develop a serverless application that demonstrated that it can be used for complex tasks like research or finding the genome engineering technology. So the way I think about serverless, again, allow me to walk you through with analogies here, is that we're all familiar with desktop compute. So on a desktop compute, you're completely flexible, you can install whatever you want, and it's very cost effective. It's like owning your own car. But it's not very scalable, and it does have a certain overhead to it. Like owning your own car, for example, you only have that one car, and if you have a larger load, you're stuck with that car. Similarly, you have to bring that car to the service, which costs time and money, which you ideally want to outsource. Outsourcing and making it more scalable could be done with a chauffeur. That chauffeur would take the car to the dealership and have it serviced, and that person can go away and come back with a bigger car if you need more load. And the analogy in the cloud space is sort of auto-scaling groups that can scale up and down with the workload. And because it's in the cloud, someone else is looking after the compute infrastructure. But like a chauffeur, auto-scaling groups can be very expensive because they're scaling up slowly and they're not instantaneously going away. And this is exactly where serverless comes in, where really the focus is on agility. So again, you have the flexibility. You do have no overhead because it's in the cloud. It is scalable, but it's also cost effective. And my analogy for that is a ride-sharing app where you can request a certain car at a certain point in time, the right kind of car at a certain point in time. And when you don't need it, it goes away instantaneous. And that exactly is how I think about serverless. Good. So with that capability of and the other interesting thing with serverless is that it's highly modular. So you can have one module that is geared for a certain genome editing mechanism and another one for another. Therefore, in my head, in the future, there will be sort of a navigation system where you can say, I want to edit this particular gene with this particular mutation or change in the end, in a particular tissue with a particular outcome and so on. So define all the parameters of it. And then a machine learning method, for example, goes in and identifies exactly the right approach, the right reagents, and the right condition in order to do that. And this is what we sort of the vision that we outlined in a paper last year in the CRISPR journal. And again, I think for that, really having scalable data structures is absolutely key. Also key is it in the human space. So here in human genomic information, where we're saying that the genomic data is growing ever larger, and therefore being able to share that data efficiently is going to be crucial going forward. So for example, in the rare genetic disease space, a patient might come into a clinical pathologist and therefore have that genome sequenced, and then there might be 200 candidate genes where legitimately there is some protein broken or there's some true pathogenic element potentially going on. And the question is, which one of those 200 options is the one that is actually driving the rare genetic disease that the patient has? And the typical approach will be then to go to other data sets in the world and say, have you seen this particular genomic mutation at that particular position? With the idea that if that mutation has been seen somewhere else, it's unlikely driving the rare genetic disease of that individual, because by definition, it is likely to be a rare change that has caused the disease. Therefore, the success of this approach really depends on more data being shared internationally, and specifically of ethnicities that are currently not very well covered. So Caucasians, of course, are very well covered, but there's a whole suite of other areas and other ethnicities that are not covered. And therefore, this whole approach of asking, have you seen this mutation? Is that mutation rare or is it actually common? Would break down for those populations. So what we developed is a more scalable approach and cheaper approach called serverless beacon. So again, it's based on the original beacon protocol, but using scalable data structures, namely cloud and serverless, in order to do that. Now, I'm not going to go through the actual architecture. Suffice to say that there's multiple Lambda functions, which is basically the compute grunt of serverless, that make this whole thing scalable. So again, a little bit of a quiz on the side. How expensive do you think it is to host 100,000 samples, and how to share that data? How expensive is it to share that data with the world per month? And this is in euro. Can we find that question? Okay, I don't think we have that question. So again, in your head, is it 10 euro? Is it 50 euro, 100 euro, 10,000 euro? Put it in your head, and I'm going to reveal that now. So traditionally, doing this, sharing that kind of data will cost you around about 4,000 euro a month. So this typically is done by the large consortia like Genomics England or the Broad Institute. They can afford to put that much money on a monthly basis in order to share data sets like that. But we brought that down to just 30 euros, 13 euros. So for less than a cup of coffee per day, you can share genomic data with the world. And therefore, the smaller research organizations around the world that might have 100,000 samples, but also might have fewer samples, they can easily and automatically share that kind of data with the world, and increasing the pool of diverse populations that can be queried internationally because of the scalable data structures, and this being so cheap. Obviously, we wanted to put this to the test of how scalable it actually is, and came up with the example of, well, we want to share a population scale data set. So there will be 350 million samples, the population of the US. And if you have the population of the US with 300 million, chances are that every single location will have been seen at least once to be mutated. So likely there will be 3 billion variants in that data set. So therefore, 3 billion times 350 million is more than a quintillion data points. And we want to process that kind of data in real time. We want to have someone on the other side of the world be able to query the population of US to say, have you seen this particular mutation and how rare or frequent is it? And with serverless beacon, you can do that, and you can do that in real time. So within a second, you can get back the results. And again, this is due to the scalable data structures and using AWS cloud computing. Again, saying this is not just for the human genomic space, but it's also interesting for infectious diseases and monitoring infectious diseases and the individual strains that are emerging around the world. So for example, with Omicron, Omicron is 86 differences to the original Wuhan strand, and 5 of those are sort of the keystone mutations that are defined now by Omicron, which means you want to go in and identify where in the world have you seen profiles with these specific 5 mutations occurring. And with serverless beacon or path beacon, in that case, you can do that. You can define any which one of profiles, of putting your own profiles together and query the data set, DIS8 data with 5 million samples around the world, and can identify where they are, where they have been, how are they spreading, can we identify the mutation or the profiles that are of concern and how they are emerging. Good. This brings me to the last story around global collaborations and how we can use modern data structures or modern infrastructure in order to deliver that more efficiently. So here I couldn't find a quote, so I made up my own. And that I think that cloud really creates this collaborative environment where we stand on the shoulders of giants by having modules and build upon modules, which generates knowledge more rapidly and of course changes the way we probably can do business. So allow me to go back to fundamentals and talk to you about how I see the cloud. So to me, the cloud is like an in-room sound system where you will bring along your tablet or your phone and you plug it in and you can listen to your kind of music in the infrastructure that the room provides. And it's akin to cloud computing where you bring along your virtual machine or your infrastructure that you want to roll out on the hardware of the public cloud provider. Depending on what kind of music you like, you want to have specialized equipment in there. You might want to have a subwoofer, or you might want to have a surround system. And it's the same with the cloud system, with the cloud setup, that you can have scalable systems, so more compute, but also specialized computes. So for example, GPUs or more, or nowadays quantum computing. And all of this you can have access to instantaneously, and you don't have to have a huge investment in the actual infrastructure yourself. And obviously we're predominantly working with AWS, but Azure and Google Cloud providers have the same concept. So again, a little quiz. Are you using public cloud computing? Oh, interesting. This is a more interesting poll in that it's very diverse. It's 50-50, in fact. Oh, very good. So hopefully this inspires you to have a little look at what cloud computing can do for you. For us, cloud computing, really, the big arguments around cloud computing is that it is cost-effective and scalable to variable workloads. So we can start off with individual prototypes and then expand that to the actual production system. Also, we can have access to the latest and newest compute, be that GPUs or be that quantum computing. And we can do that without having an upfront cost investment in the hardware ourselves. But I think the biggest argument for us for the cloud is that we can interact with the world. We can develop something on our local cloud that can then be rolled out on the other side of the world without any lag time and without any changes, because it's the same infrastructure that is used. So as I was saying, we're working together with AWS, and we had a partnership with them for the last 5 years, where in 2016, we developed the first serverless application to really demonstrate that the concept of serverless is actually extending to something as complex as research or genomic research. In 2019, we were the first to put a product on the marketplace, and I'm going to go into a little bit more detail in a couple of slides. And in 2020, I became a data hero, or AWS hero, specifically a data hero. And I'm currently the only one from a non-IT academic background. So if you're working in the cloud and there's something that AWS is not quite doing right for you, so please reach out to me, and together we can sort of help AWS cater better for the academic research and innovation space. Good. With that, coming to the AWS marketplace. So no surprise, we put Variant Spark as probably our most modular and most advanced product on the AWS marketplace. And here the benefit is that everything is wrapped up into this one deployment element, infrastructure as code. So that is the actual algorithm, the workflow of how to best practice, analyze genomic data, but also how to keep the data encrypted and how to do the management of the individuals that interacting with the notebook style analysis. And all of that is packaged up, is available on the AWS marketplace currently for free. So you can just press a button, it automatically spins up the entire system in your account, and all you need to do is point it to your S3 bucket with your genomic data in it. Now with this, obviously the idea is that you can roll out the same analysis, and that I think is the personification, if you want, of reproducible research, because it's on the exact same hardware, it's the exact same workflow, and it's the exact same data or data that you would point it to. And therefore, you don't have to have this traditional overhead of thinking about how to install something. The libraries are not compatible, or the infrastructure, there's some quirk there. The data is somewhere else and you can't get it. With this one, everything is in one package, and it's really just a one button deployment. And I think, to me, this is how research should be and how we can together evolve the field forward more rapidly. Good. With that, the three things to remember is efficient algorithms are really key going forward, because even with cloud computing and more compute power available, having an algorithm that utilizes this infrastructure efficiently is going to be key, because the data is only getting bigger. For example, with Variant Spark, we developed an algorithm that can vertically partition the data, random forest, and therefore can compute the random forest in parallel by vertical partitioning the data. And this helps identify disease genes, specifically complex epistatic interactions in large cohorts. Also, it will be important to have clever and scalable data structures going forward that allow modular approaches. Again, unprecedented data volumes and having to collaborate more in the future. These individual systems that you can build upon are going to be critical. And my example here was GT-Scan for genome engineering, where we have individual packages for the individual genome engineering tasks, or serverless Beacon, which allows to efficiently look up or exchange genomic data, be that for human rare disease research or for COVID or other infectious diseases, for tracking them around the world. But most importantly, I think it's absolutely crucial to have global collaborations, to really work together in order to build individual tasks that are larger than the sum of their parts. And for me, the public cloud provider marketplaces are really a good way of getting started with that, because here you can put everything into one deployable element that is going to use the same infrastructure, the same hardware, and all you need to do is press a button. You bring up the infrastructure that was, for example, published in the paper, and then from there you can improve and build upon your own solution. Good. With that, I have one last quiz around which one was your favorite part. Was it the algorithms? Was it the scalable data structures, or the idea of needing to work together more? With that, thank you very much for listening. And I think we do have some time for questions. Oh, sorry. You probably want me to read out the first question. The first question is from Tuk. Sorry if I pronounce your name wrong. Is the first algorithm approach that uses the data to produce a GWAS applicable for non-model species? Yes, that's a very good question. I think the biggest problem here is that, so in principle, yes, because it's fairly agnostic of the actual application. We can use it in human, we can use it in COVID, right? I think the biggest problem is to identify the individual mutations that a non-model organism might have, because this is typically done by aligning it to a reference genome and therefore having the mutations identified through that. But say, for example, if you have a SNP array where you have already individual locations and you have that with your data, then you do get the outcome directly, and therefore the organism itself does not have to be as characterized. And that data you can feed into VariantSpark as well. So I think in principle, yes, the question is then more, how do you actually interpret the data? So if VariantSpark comes up and says SNP 5,250,000 and 7,223 work together, and you don't know what kind of genes they are in because there is no information for that genome, that makes it probably tricky. But yeah, very interested in in seeing how it would work together. So again, get in touch with me.
Any other questions? You may vocally or in written form ask them.
Okay, while we wait for questions, and while I still have the poll open, are you using public cloud computing? So I think the final one of 37 people who voted is that 51 have used cloud computing and 49 have not. I think that's a quite interesting result, in that probably only last year, it probably would have been much lower, in terms of how many people have used cloud computing. So I think there's definitely a trend in recognizing that there is this large volume of compute power that we can just easily use. But also, I think there is a realization that to some extent, sovereign capability and sovereign high-performance compute facility do have their value as well. At least, that's what I'm realizing more and more, in that there is this beauty of working together globally through the public cloud providers. But at the same time, we do need to have the capability of doing our analysis in situations like COVID, where, for example, the world is shutting down and suddenly you are on your own. And especially here in Australia, we were very much on our own. And therefore, I think hybrid is going to be where the future is leading, in that you outsource some elements to the cheap, easy scale, global collaboration, but you do retain some sort of core capability within your own realm, so you can compute here and share the data and share the results internationally.
Dr. Bauer, I think we have a couple of more questions. The next one is, do you think cloud computing is safe for storage?
Yes, again, very good question. So there are two elements to that. Technically, it's probably the best storage you can think of in terms of keeping the data safe. Like, I think it has to the power of, no, they have 9 zeros in terms of the dropout rate. So 0 point, and then I think there are 9 zeros for the dropout rate. So it's very secure because it's replicated in multiple regions. And even the regions have their own independent data centers. So if one data center gets hit by a meteorite, you know, the other ones are still up and running. Plus, you have this whole team of experts that have nothing better to do than think about how to keep the data safe there. In saying that, obviously, there's an element to it that if you put it in the cloud, there's some considerations around how to keep it safe, where before it was security through obscurity because it was on some hard drive somewhere. Now you have to actually put active security in that place in order to encrypt the data, keep it safe, so it doesn't become a honey trap where people can hack your account. But again, because there are these large groups in the public cloud providers that have nothing to do better to do than making that approach easy, it is now just a press of a button to keep your data secure and safe, in saying that it needs to be encrypted at rest, it needs to be encrypted at transit, and the keys are located somewhere else so that these two things are truly separate. So therefore, even if your account gets hacked, your data cannot be read out because they're encrypted. So there are now these really best practices around it that are easy to follow and easy to implement to keep the data safe on an international standard.
Okay.
Osman, could
yes.
I ask a question?
Of course.
Hi, Denis, how are you?
Hi,
good to have you. And again, as usual, excellent talk. And I did speak to you sometime back in 2019 about collaborating, and I hope this revives that conversation. And I look forward to approaching you soon to see how we can take that forward. My question is with regards to cloud, because we just recently set up our own server here, nothing fancy, but at least something to get started. The concern with cloud is the hidden cost. Of course, one aspect is sending the data. If the provider is not within your country, then if you have a lot of data, then you gotta ship the hard disk to them. They'll probably upload it for you. So that's one, the time it takes to do that. Secondly, when you want to download, I think the cost of downloading the data eventually is much higher. And then thirdly,
Oh yeah, I think we lost you.
Oh,
yeah. What was the third point?
Yeah, the third point is, you don't really know, let's say you don't really know your demand or how much you will use. If you're using occasionally, maybe it's okay, but if you're going to use on a regular basis, I think the cost there would add up. I know people who have like 10,000, allowed quotas or something, and then they just run out of that very quickly. So what's your advice? What's your take on the cost aspect of this? And I think that's one of the fears, one of the concerns, and people then still invest in having their own in-house servers.
Yeah, absolutely. And this is something that I keep telling AWS, and probably if more people would tell them, that would be that would be good. In that, I currently think that a lot of people are held back by that fear of accumulating this huge bill that cannot be managed. Because as researchers, we're not typically, especially in domain researchers, we're not IT experts. And therefore, this whole notion of needing to develop the perfect architecture from the beginning scares a lot of people, including us. It did scare us as well, which is a fair point. So therefore, if AWS listens to that to some extent, in that there's a lot of credits available and there's a lot of flexibility around the actual cost for research, not for industry, but for research, there's a certain flexibility around of catering to that, to the specifics like domain or lack of domain expert, if you want, for researchers in the IT space. Now, saying that, the way we do this is that we start off small, where we know that even if we get it wrong, it doesn't completely escalate into a ginormous bill. And then we slowly dial up the heat and making the data sets bigger and the runs longer and so on. So I agree with you. And this is a learning curve that you do need to think about which infrastructure is the best for you. You can't just simply spin up an EC2 instance as your first port of call. For example, it sometimes needs to be a serverless infrastructure where the bursts that are running, where you're only temporarily running the analysis, and then it goes away to cost nothing when you're actually not using that. Whereas for an EC2 instance, you do have to pay a certain amount just to keep it live. So I hear you, and I agree with you that AWS needs to do a little bit more to support us non-IT experts, bioinformatics and so on, but not IT experts, in order to do our research with a little bit less anxiety.
Thank you, Denise.
Hi, I have a question, may I?
Yeah, go ahead.
Of course, yes.
Hi, Dennis, great talk. I'm really all for cloud computing and stuff. I'm just wondering about especially with the COVID and stuff. I believe there is the cloud computing services are morely centralized or controlled by some certain groups. And I'm wondering if there is any work against towards the decentralization of this cloud computing in the whole world, especially with this COVID stuff happening. I'm just curious if there's any work towards that.
Great question. I'm a firm believer in decentralization and federated data, decentralized identity and so on. So I'm definitely in that camp. The problem I think here is that economy of scale only works when you have a collection of experts that can bring in the economy of scale, right? That create large data centers, know how to do that, and then just replicate that around the world. So I think the cost saving that we're currently seeing, we can only ever achieve if there's one corporation or a set of corporations centralizing that around the world, unfortunately. Therefore, I think multi-cloud is absolutely crucial in order to curb that, that a tiny bit more. Now, I do know that the idea of the open cloud is something that was attempted 2 or 3 years, that's sort of my knowledge stand, 2 or 3 years ago, where sort of the idea was that all the high performance compute facilities around the world contribute their data or sign up their resources in a open cloud distributed capability. I'm not entirely sure how, where they've gotten to, because again, it comes to that you probably not have this coordinated approach of what a smooth operation would deliver. So in research, it's probably not as crucial if the thing breaks down and you would have to start from scratch again and do your analysis again. Like the uptake, the data security, and so on is probably not quite as crucial as in the commercial space. In saying that, I mean, we don't want to lose our data either. So yeah, I think these are the concerns really, or the two competing interests, in that on one hand, you want to have this smooth operation, which you probably only get from the economy of scale of centralizing it. On the other hand, decentralization is a great idea. I'm totally for it. And the open cloud initiative is going in that direction, but I'm not sure how much headway they already made.
Thanks a lot, yeah. With the COVID from NVIDIA to BMW, all these sectors, they were opening up their resources, and I thought like finally we are going to decentralize and have a great resources in a cloud environment. But I understood. All right, thanks a lot for your thought.
Yeah, look, I'm still optimistic though, in that with all the decentralized identities and decentralized finance and those sectors that are popping up on this concept of decentralization, I think whatever protocols they develop is translatable directly to the cloud computing space. And therefore, I think it's probably only a matter of time before we have some sort of stable thing over there.
Okay, I think that ends our session. Thank you very much, Dr. Bauer, for a wonderful talk. There are still a couple of questions outstanding. Maybe they can contact you offline and you can address them. All right, thank you everybody for attending, and thank you again, Dr. Bauer.
Thank you so much. Pleasure to be here. And definitely get in touch with me either via Twitter or email. Talk to you soon. Bye.
Bye.
So thank you, Dr. Osman and our honorable keynote speaker. It was an excellent presentation. I hope everyone here has been enlightened by Dr. Denis' talk. Now it's time for our SG Turkey led session. I now invite Mr. Batuhan Çakır for his presentation.
Thank you very much. Now I'm trying to share my screen. Can you see my screen?
Yes, we see.
Okay. Hello, everyone. I'm Batuhan Çakır. I was secretary of RSG Turkey this year. And before starting, I would like to thank to event organizers who asked us to support this beautiful symposium. And we are very glad to do that. I would like to thank on behalf of RSG Turkey. And with this presentation, I would like to introduce you the RSG Turkey. You might hear our community or not. I would like you to be familiarized with RSG Turkey. So what is RSG Turkey exactly? Actually, we have a longer name, ISCB-SC RSG Turkey. We are a student group under International Society for Computational Biology. This society is based on the USA, and this society has ISCB Student Council, which has a bunch of regional student groups in different countries. And RSG Turkey is one of these groups. And our group was founded in 2011, and this year we celebrate our 10th year, and we are very happy about that. And this group was initiated in 2011 by some assistant professors, and since 2011 we have some in previous we had some leaders, as you can see here. And as RSG Turkey, we organized many events. For example, in previous years we were organizing face-to-face symposiums. This photo is from our last face-to-face symposium in 2019 in Izmir Biomedicine and Genome Center. After this event, we needed to organize our events online because of the COVID. And we also organize webinars. Until now, we have organized more than 50 webinars. We had more than 1,000 attendees from 40 countries. And we have more than 300 members on our BioInfoNet channel, which was founded by the founders of RSG Turkey. We use our Big Marker platform to organize our webinars. We have a BioInfoNet channel under this platform. And We also translate the English webinars into Turkish to make them more available to other people. We organize different kinds of webinars, like webinar series specifically on population genetics, and we also organize student webinars. Yeah, as I said, our webinars are being translated in Turkish. And in our website, we have a section called resources, and we collect some useful resources about computational biology. We try to keep updated this resources page, and it's an open source page. And besides of that, we have some blog posts shared by our active members about many different topics. And also we are open to anyone who wants to share their contents on our website. And we are also organizing workshops. This workshop, Occupy, was in 2019 to teach Python. And also each year with our symposiums, we also have workshop sessions. In previous years, we had workshop sessions to introduce R, also machine learning with R, and data visualization by using ggplot. And we also organize some separate workshops separate from the symposium, like this one. Last year, we organized single cell workshop to introduce people single cell RNA-seq and single cell attack-seq. And we are also organizing some panels as a live stream on YouTube. For example, we organized the live streams panel about AlphaFold, and we had another panel about to discuss with high school students who were working on SARS-CoV-2 virus by using machine learning techniques. And besides of that, we have organized some career fairs to discuss their current and previous experiences. And also we have organized some Instagram live sessions to discuss where and what do bioinformaticians do. And we are organizing journal clubs each month, and we are planning to keep doing that. And we believe that RSG Turkey is an excellent community to be part of. If you would like to be the part of the RSG Turkey, I would like you suggest to register from our website. We are active on Twitter, Instagram, Facebook, YouTube, and LinkedIn. By using this link and this QR code, you can access our all social media accounts and link for our official website. And from our website, you can register to our RSG Turkey community, and then you will get an email about joining our Slack workspace, which we use as a main communication tool. And before moving to the panel section, I would like you to announce that we will organize a computational structural biology virtual workshop tomorrow. If you're interested, I'd like to suggest you to register from our website. And as I said in the previous slides, we keep organizing our journal clubs. We have the last one in next Saturday, and we will organize many more journal clubs in 2022. And we can move to the panel discussion session. In this panel session, we are planning to discuss the current topics in computational biology. Our guests for today's panel is Dr. Beste Turanli. She is an assistant professor in Marmara University, Department of Bioengineering. Dr. Ezgi Karaca is an assistant professor in İzmir Biomedicine and Genome Center. And Dr. Tunca Dogan is an associate professor in Hacettepe University, Department of Computer and Artificial Intelligence Engineering. Before starting to ask my questions, I will kindly remind you that due to the time limitation, I will kindly ask you to limit your answers to 2 minutes only. And I think we can start our questions. So
actually, there is one question on chat. If you want to answer before we start
yes,
our panel session.
Sorry.
So can I can ask for you if you want.
Okay.
So is RSG Turkey open to non-Turkey based, non-Turkish?
Yes, we are open to anyone. You don't have to be Turkish to register to RSG Turkey. We are open to everyone.
Okay, so there is one comment as well. I'm interested to if RSG is open to non-Turkish. So
yeah, we are definitely open to non-Turkish people.
Okay.
Don't hesitate to join us.
Okay, so the floor is yours for panel session as well. Okay.
Okay, thank you very much. And I'm starting with my first question. How did you get into the field? And do you think if there is a best background for bioinformatics? I think we can go with alphabetical order. Dr. Beste Turanli.
Okay, then. By the way, before I start, I would like to thank, because it's a pleasure to be here, and it's an honor to be invited by RSG Turkey for me, together with Tunca Dogan and Ezgi Karaca for sure, because I always admire them. And, you know, I feel that maybe I am the new beginning, like a junior in this group. So how I started bioinformatics, actually, I was a wet lab person in my bachelor and master. Then I shifted bioinformatics in my PhD. So I always wanted to work on medical biotechnology, but I didn't decide the tool exactly. Then I decided to choose my supervisor. Actually, it was a little bit emotional choice, but that's the reason that I know myself and I'm people-oriented. If my coworkers are positive, motivated, and hardworking, then I became more, like productive person and then happy there. So it was my driving force. And I deeply believe that there is no special background. So that if you are a physicist, chemist, biochemist, bioengineer, whatever you are, I mean, if you have enough motivation, then you can work hard enough. You can, maybe it may take time to get all your goals in bioinformatics field, but in any way you can do it. And there is no exact time to start working on it. It's my experience, at least.
Thank you very much, Dr. Ezgi Karaca.
Thanks also from my end for the invitation. So it's always a pleasure to meet with RSG Turkey's occasions. And also thank you, Beste, for the kind words. So I actually agree with Beste as well. I don't think that there is an optimal starting point to enter bioinformatics. It's like for many fields it's like this, but I think for especially in bioinformatics, it's more like as long as you like what you do, you're going to learn it and you're going to be successful in it. So the only thing for me is like whether you like working with a computer, but not like, I don't know, like you sitting in front of the computer for a couple of hours using Word and PowerPoint. That's not it. But really, it's not that healthy, but still you have to do it. So whether you're okay with sitting in front of the computer for long hours, and whether you're okay with working with conceptual work, conceptual studies, meaning that you're not gonna validate things maybe with experiments. So it's a different type of hands-on technique. But it could be also the best choice for you if you like this type of work. For example, for me, that was it. By training, that was already a long time back, but I graduated from chemical engineering department. So I can confidently say that any engineering department is suitable to enter bioinformatics field, because this is what you learn in engineering. You learn to conceptualize things in theory and also in practice. And besides this, I think, as I said, like whether you kind of feel familiar with this concept, with working with computers, etc., that should be a sort of criteria to decide whether to enter this field or not.
Thank you. Dr. Tunca.
Okay, first of all, thank you very much for the invitation. It's always nice to be with you in these panels in the different types of organizations with RSG Turkey group. And also I would like to thank Beste as well for her nice words. So speaking about when to enter this field, in my experience, I was a student in engineering, actually, engineering faculty. I graduated from process engineering, and now most of my friends has been working in companies in private sector as production engineers or R&D engineers, and less of them has switched to the academia actually. And during my master's degree, I switched my field a little bit to the molecular biology and microbiology, because my supervisor was doing this actually in her lab. And then during PhD, I switched to bioinformatics. So it was kind of late for me, I guess. I think it would be better if, of course, if you are doing first master's then doctorate, then the students, if they can start at the beginning of master's, switch to the field, then it would be fine. I think it's not very necessary to have a bachelor's degree in bioinformatics. Anyway, only 1-2 universities have these degrees. So I think it's good to have a background from chemical engineering, computer science, statistics, also molecular biology, and even chemistry is fine, because you might be aware of that many bioinformatics applications are switching to the field of chemistry, and a new field has born. It's not famous as bioinformatics, but it's called cheminformatics as well. So it's also fine. But I would recommend for the people who would like to go into this field to take at least some courses. There can be courses from their university if they are available, or they can take some courses from online sources. Like there are many different companies that are offering free courses as well, to learn some sort of programming, at least if they are coming from the biology background, especially fundamental sciences background, during their bachelor's studentship, okay, at their 3rd or 4th year. And then they can easily switch to the field of bioinformatics during master's or doctorate.
Thank you very much. Now I would like to move to the second question. And many biologists now learn core bioinformatics skills in their undergrad. Do you see having only a computational background as a wise choice, or do you suggest learning wet lab skills? What would you suggest, Dr. Beste?
So again, I will start with my experience. Actually, I encourage learning wet lab skills. So, but please keep in your mind that when I say this, I mean learning in theories, far from the practice in the lab. So I suggest only learning in theory for the one who has only computer background. And I give compulsory courses named Introduction to Modern Biology for the last 2 years in Marmara University Computer Engineering Department. And there are many enthusiastic young friends to learn biology. And I'm so glad to hear their motivation. And maybe it's because that every single time when I start the course, I am asking them, I am I asked them to, do you know that Alan Turing wrote a biology paper? So then said that no, because they're always thinking that he is the pure computer scientist. It's like a father of this programming and algorithms. And then they changed their mind to start to get involved biology a little bit. So it's also the same for the guys who has a biology background. So now it's integrated. So if you know biology, you should learn bioinformatics skills, but every single time you also know how the data created. That's why it's important, the wet lab skills, the techniques, how we are sequencing the data at least. The basic theory is always helps you to understand the data for this informatic practices as well. It's my opinion.
Thank you very much. Dr. Ezgi Karaca.
So when we say bioinformatics, we don't talk about a single discipline, actually. It covers many different subdisciplines. For example, the type of research we do is in the field of structural biology. So if you are in the field of structural biology, for example, if I would say like, okay, if I would advise the students to learn how to perform structural biology experiments, that would be way out of scope, because just learning the type of experiments these researchers do might take 4 or 5 years to really conquer it, to really know it. So that's why I think like discussing these things in general terms might be also not very beneficial. That's why, for example, the example that Beste gave was very good. Like, if you know how the sequencing technique works, then you know where the noise comes from when you analyze your data, etc. But these, for example, these type of experiments are going to be performed in a way and more easy manner. They will be more and more in a like a packaged content that the experimenters will have less and less contribution. So what I mean is, like, also on the experimental side, there are experiments which are going to require least amount of human effort, and still experiments which will require a lot of human effort, like biochemistry experiments where you purify proteins, etc. So in that sense, I think it's also important in which specific field you are working. But anyhow, irrespective of this, I think it's very important to know the type of data, the background of the data that you are using, so where the data is coming from, how to interpret the data. And to be able to understand this, I think the best way is to collaborate with experimentalists. And if you have the opportunity maybe to go to their lab, to visualize what they do, how much time it takes to perform their experiments, etc., etc. So that would be my initial advice. And then, depending on the type of experiments that you are going to analyze in your computational world, then I might advise, okay, go and try something hands-on. But in the case of, as I said, like structural biology, I think that would be out of scope, because these experiments are very tedious and also very difficult to learn, and that should be left to the experts.
Thank you very much. Dr. Tunca.
Okay, thank you. I definitely agree with both Beste and Ezgi. So they summarized it very well. I think at least I can say that wet lab skills is something different from theoretical knowledge about the field. Theoretical knowledge and what has been done in the literature, keeping up with the literature, these are quite important things. And I definitely recommend people going into the field of bioinformatics and computational biology, especially coming from a computational background. They should definitely keep up with this, but on the other side, wet lab skills is something else entirely. So it would be like saying people going into the bioinformatics and coming from biology backgrounds to know how to build an operating system, for example. You don't have to know that, of course, but you use operating system all the time, but you are a user. So this is something similar to that.
Thank you very much. Now I would like to move on to the third question. Recently, machine learning and deep learning started to be used in studies of structural biology. How do you evaluate the effects of machine learning, deep learning into the research? Dr. Beste Turanli.
Okay, so this question is a bit tricky to me because neither machine learning nor structural biology is my expertise. So probably Ezgi and Tunca will answer better than me. But I can try to answer the perspective of what I'm studying now. So I'm trying to do multi-omic data integration. So it's basically network science that I'm studying now. And when I check the data that I have, there are diverse data, including like molecular expression profiles, physiological measurements, medical imagings. There are many clinic, environmental relevant information that all needs to be integrated to get a real life conditions to see what's happening now. And it has many dimensions. It spans many dimensions. So to cover all these topics by a conventional, this computational approach, it's not so, it's not easy to do that. That's why artificial intelligence, machine learning, deep learning techniques will help us in the future. So I can say that it's encouraging for us to learn artificial intelligence algorithms as well to manipulate and integrate all the data. At least I can say it from my perspective, what I'm trying to do. So for the structural biology, now I cannot imagine what's going to happen in the future, actually.
Thank you. Dr. Ezgi Karaca.
So I think, yeah, indeed, we are going through a very exciting time. Some things are going to get obsolete in a very short amount of time, thanks to the advancements in AI, in computational structural biology. So this I consider as a very positive improvement. I'm really happy to see that there is enough data accumulated in the literature so that these algorithms can do something with the data that is available, and that's going to be useful to us. And this is going to accelerate our research immensely. And some things that we have been thinking that, okay, we should wait 10 years to answer these questions, we'll be able to answer them maybe in 1 or 2 years of time. So on one hand, of course, this also puts a huge pressure on the shoulders of people working in the fields where AI is becoming more and more logical and useful to use. Because if you're not an expert of AI and you want to understand what's happening in your field, then that could be a bit stressful to you. But on the other hand, I think this also creates a pressure to start a different collaborating atmosphere. So for many years, and this is how it should have been, we collaborated with experimentalists. But now the computational people will start collaborating among each other. I'm not saying that this wasn't happening, but I think it's going to happen more and more frequently. So the computational people will gather bigger teams to address more challenging biological problems, and I think that's going to be fun. And this is what hopefully we will be achieving also together with Tunca.
Thank you very much. And Dr. Tunca Dogan, how do you evaluate the effects of machine learning, deep learning into the research?
Yeah, okay, thank you. I agree with Beste and Ezgi again, and I should add that so this has been the applications of machine learning and deep learning has been associated with structural biology, mostly because media picked up the success of DeepMind due to the production, their development of AlphaFold. But before that, machine learning has been in use both in structural biology and other fields that is related to computational biology and bioinformatics for many years. So you can, if you take a look at the literature, you can find some papers in the late 1990s talking about an application of at least not deep learning, but classical sorts of algorithms, shallow algorithms for solving problems related to biology. Okay, but since media is interested only in these kind of huge achievements, and also in terms of artificial intelligence, which is a quite popular topic, and DeepMind is producing this, they only pick this up and show it to the people. And if you are not an expert, of course, only thing that you would know is that deep learning has been in use for structural biology by DeepMind. But we've been working on these topics many years before. In terms of specifically for deep learning, it started in like 2015 and so on. There are many studies, projects, papers that came out of this kind of work. So it's not only structural biology, but in many, many other fields. But of course, in structural biology, there has been huge achievements as well. Again, not only by AlphaFold and DeepMind, but many different universities and research groups, such as the Baker Lab, who developed Rosetta Fold. And I think in the future, the AI will have a penetration into the field of structural biology and molecular biology and genetics and other specific fields of life sciences in the future to solve the problems that we are working on. We can, of course, talk about this for many hours, but since our time is limited, I should stop at this point.
Thank you very much. Now I would like to move on to the next question. As you know, biological data is increasing day by day, and it's getting harder to analyze and store the data. The easy transfer and management of biological data have recently become a topic of great interest. How do you think bioinformatics can find a solution to this in the future? And is there any approach or study that you know of? We can start with Dr. Beste.
Okay, thank you for these questions again. Who chose them? So it's very nice. So what we are talking now is like, okay, the biological data is increasing day by day, but on the other hand, there are different opportunities like cloud computing. Dr. Denis explained it very well. And she also explained the serverless systems. So we can store all the data or manipulate the data. So we can also do whatever we want. So this transforming the data or storing all other things, all the things that we would like to do now is available by cloud computing now. And I believe that time by time, while we are accumulating the data, probably we're going to also accumulate the information about how we can manage all these things. And again, it's coming to the bioinformatics field to create a new efficient algorithms. Like one goal can be like compress the data form to make it easier to storage, for instance. Probably bioinformatics will be there to create new efficient algorithms. So there are also other examples like Nick Goldman's DNA storage. So we can also create some other alternatives to store the data. But I think now we're going to use cloud computing because it will take time to store all the information into DNA, because in any way, as I remember, it started like maybe 5-6 years ago, and they still, okay, they are doing this. Probably they were, as I remember, they were working with Agilent as a company. So it's so expensive to achieve. So for our future, yeah, it's so promising. But probably in today's world, in the short time period, I can say that cloud computing will be the only solution for us. And the algorithms and the methods that we created as a bioinformatician can help us to store the data or manipulate or transfer.
Thank you very much. Dr. Ezgi Karaca, how do you think bioinformatics can find a solution to this data storage problem in the future?
First of all, I don't think that this is a problem only for our fields. So how does Facebook store all these pixel information, for example, or I don't know, Instagram? You know, So I think it's a global issue, not specific to our research field. So the best would be probably to check these other fields to see what they do and how the type of encoding algorithms they use can be implemented in our field. So that could be an idea. So that's one thing. And regarding, cloud computing, I think it's a great option, but it's a great option for developing countries like us, as long as you use a state-funded resource, like your national cloud, or if you are a member of any consortia, then the cloud of that consortium, for example. That could be an idea. Because if you want to use Amazon Cloud or any, doesn't matter, the name doesn't matter, so any kind of private cloud service, you are basically bound to their pricing, and that's usually very expensive for many countries. So that's why I think if you want to go for cloud computing, you should also look for the opportunities, like, do you have a national cloud source, or do you have, is your country a member of specific consortium where you can apply for cloud storage or cloud computing time, etc. So that would be my second thing. And second point for regarding this point. And as third, I think we should more and more start to be aware of the fact that whatever data we produce, we generate, we use, we process, this is going to cost electricity, and that will have some environmental impact. So that's something that's not being very heavily discussed nowadays, but I think it's going to be the case in a very soon time. So whatever we store in our Google Drive, or whatever intermediary or local mean that we use, I think we should be very mindful about the data that we store and that we use, because, as I said, any storage or any processing that might not be that environmental friendly. So what we do is not for free, although people think that it's for free, but it has a cost on the environment as well. So that's the third thing. So combining all together, I'm not sure whether we are the right experts to be able to find the solution to this. Probably we should consult computer science people about this. But in the end, the best thing would be just to check the other disciplines or other markets to see how they're dealing with this issue, and maybe try to find a solution for our own field as well.
Thank you very much. Dr. Tunca Dogan, how do you think bioinformatics can find a solution to this problem in the future? And is there any approach or study that you know of?
Okay. Actually, I agree with Ezgi. This is not specifically a problem of our field, bioinformatics or computational biology, because this is a problem related to our era, actually. We are living in the era of data, and of course, we are producing data all the time, and it should be stored and transferred somehow in an efficient way. There are many concerns, ethical concerns or environmental concerns, and they are being taken into account to, apply new policies. And governments, especially developed countries, are dealing with these situations, start to deal with these situations now. Of course, in our field, we have the similar problems, and we are going to follow these fields to, find some applications for us, some scalable and efficient solutions, and then apply them to solve this problem in our field. So since this is not specific to our field, I don't think that we need to work on this. But I can make another point here, actually, that is specific to our field. This is the data security. Of course, the data security is an important and general problem, but especially patient-specific data security is an important problem, important issue. And people in the field of bioinformatics and computational biology are working on this. There are people, there are research groups, even in Turkey. If you're interested in this topic, you can make a search and you can find some group leaders that are doing fine work on this topic. And especially how to store this data, how to share this data, because stakeholders should have this data and you need to share the data so that people can work on it, but it should be anonymized somehow, and the ethical concerns should be taken into account. And this is all a different field. So there are many projects, papers that came out discussing this problem. Maybe people who are interested can take a look at them.
Thank you very much. Our next question is about cloud computing, and three of you already partially answered this question, but I would like to ask anyway. Dr. Beste Turanli, how do you think it can be better how do you think the cloud computing can be better integrated with bioinformatics? Do you think that it will turn into something that people often prefer in future works, or do you often use in your projects?
Okay, so to be honest, I don't have enough experience with the cloud computing, because in Sweden, when I was in Sweden, we were using cloud computing for storing the data and then reaching the server and do some prior analysis. Then we were switching our own working station. And in Turkey, there is an in-house server at the department, so if we need to use. So that's why I couldn't get involved cloud computing a lot. And so I was just driving my own car, like Dr. Denis mentioned, that I was using my own working station for each of my works generally. But now I feel that I deeply believe that life pushed me there soon, because cloud computing also creates a collaborative working environment. So it's also nice to get involved there, even for the national research groups, the collaboration in the national research groups, or maybe in the international ones. So it's only way to transfer the data or share the analysis and the results probably in our near future. That's why I'm not so experienced now, but I deeply believe that I need to learn it and involve cloud computing system into my projects in the future soon. I know that TÜBİTAK also has a special cloud computing offering through the Bilgem. So let's see what's going to happen.
Thank you very much. Dr. Ezgi Karaca, do you think that cloud computing will turn into something that people often prefer in the future works?
Yeah, probably yes. It's gonna be, so maybe again, in this case, I'll use an analogy. So earlier, to stream a movie, no, even before that, we had to get the movie, download the movie. Then everybody's downloading the same movie on their own computer, like I don't know how many people. And then afterwards, that's kind of the cloud computing of TV series or movies. Then Netflix type of services appeared. And now nobody is downloading any movie or whatsoever to their own computers. So everybody's live streaming or streaming something that is already being put in the cloud at his or her own timeline. So this is kind of what's going to happen, I guess, in any data storage related field in the future, because if we store the same data over and over again, this is not very logical from different perspectives. So in that sense, having, for example, servers like from EBI or NCBI or PDB, etc. So this is a sort of cloud storage as well. So they store the data somewhere and we reach it via a web service. So there will be more and more resources like this, centralized resources like this in a soon time. And I think this can be also considered as cloud, not computing, could be also computing, but initially cloud storage. But besides this, so the storage can be solved like this. So to have more centralized resources that are being provided by the countries, like national or international ones. But so the storage could be solved like this maybe, but the computing, for computing, I think for some more time, we will have local computing. We will keep continuing to have local compute resources as well, like compute resources in-house, because not all the things are worth to submit to cloud. So you want to do some preliminary tests, or you want to do some preliminary model building, etc. So for these things, I think like to generate something testable, I think already for some more time, we will continue using our own local resources. But it would be probably in the future, like we will compute like small things or testable things in-house. And then for big things, we will go and use the grid resource or cloud resource, whatever it's going to be, whatever name it's going to have. This is also logical from funding perspective for countries, because the countries don't want to fund individual computers anymore. Instead, they make big investment on big compute infrastructures. So I guess all in all, I think there's not going to be a single solution, but multiple solutions, and a majority of the solutions will use cloud services.
Thank you very much. Dr. Tunca Dogan, do you often use cloud computing in your projects, or would you use it in the future?
Yeah, of course, we use cloud computing, and it's becoming more common every day. It's better in terms of efficiency, and also there might be some problems with your local servers and local computers. Then cloud is more, safer. At least not 100% safe, but safer in terms of retrieving your data, because these centers that offer cloud storage and computing services, they divide their centers in different geographical locations, and they store the data in multiple locations. So if something happens like a natural disaster in one of them, then data can be retrieved from the other one. So it's better in terms of efficiency and reaching your data, of course. And I think in the future it will be more commonly used. Today we need to pay some sort of money for cloud computing and cloud storage, but it will change in my idea, and this is the projection of the specialists as well, and it will become a natural part of our life. So if you go back some years ago, when electronic emails first came out, then the companies charged people to have their services, because we didn't have Hotmail or Gmail at that time. So those companies were charging like $20, $30 per month, so that the people can use electronic emails, can have an account, electronic email account. But afterwards it changed. Now we think that our email accounts are natural part of our life. So it will be like that in the future for cloud computing and storage as well. But of course, we need to have some technological developments first, because they need to reduce their costs. That's my idea.
Thank you very much. Before continuing to our questions, we have some questions in the chat section. Dr. Aisha Fatima and Dr. Mohammed Golam are asking, how high is learning curve for wet lab people to get started with bioinformatics, or how does the learning curve compare to people coming from physics, maths, statistics, and not necessarily computer science? Dr. Beste, Turanli, you mentioned that you have a background of from wet lab. What do you think about this learning curve?
It's so personal again, but still, you know, I think it's only about how you think. As I can say that, okay, I have a wet lab background, but I graduated from engineering department, and it was bioengineering, and still I was getting involved all these engineering concepts, and then thinking analytically was part of my studies as well. So someone muted me. Okay. So for me, it was easy to adapt, but I don't know for a pure biologist, how can it's possible? But probably, I know Melike, probably she is also graduated from biology department, right? And she is doing well. So it's still so personal, but I don't know how to answer this because I see the results from both sides. I know the people who cannot adapt themselves easily, so the curve is, going so low levels. But I also know the people who can easily adapt themselves and create great works.
Yes, thank you very much. Dr. Ezgi Karaca, do you have any thoughts about this learning curve?
So I had and also have students from microbiology and genetics. I am actually a faculty in an MBG department. And I teach in this department Introduction to Bioinformatics or Advanced Bioinformatics, etc. So I have a sort of feeling of how microbiologists are prone to learn technical aspects of bioinformatics. So I can say that the initial phases of the learning curve is slow, but then it gets faster. So from my experience, I think for a year, you should just do your best to learn as much as you can. And you shouldn't get demoralized if you don't feel like you're advancing. So I think a year is that could be interpreted as a sort of preparatory year to enter a new field. But then after that first year, things start going much faster. But we could also take this from the other way around and think like, okay, how long would it take for a computer engineer to understand, to have a feeling of the biological data that he or she is using? So there is a learning curve there as well. And that is, I think, in some circumstances that goes slower than goes up slower compared to a situation where a biologist is trying to learn programming. So it's personal on one hand, but on the other hand, it's also related to exposure. So even, it doesn't matter whether you are a biology graduate or not, everybody has a level of exposure to programming or to computers, etc., etc. But the other way around is not usually the case, like having an exposure to biology for a computer science person. So I guess everybody coming from each discipline is going through a different type of difficulty, but in the end, this is what grows you. So you should be patient until you feel competent enough to have some progress.
Thank you very much. Dr. Tunca Dogan, what do you think about learning curve from different fields?
Okay, yeah, I agree with Ezgi while saying that there are both sides of this situation, so that people coming from the computer science background had to learn the biology as well. And since this exposure is very limited beforehand, then it would be harder for them to get into the field. And also, in my personal experience, people from the biology background, and we can say life sciences background, they are willing to learn the computational side of it. They are very eager and motivated, but people from computer science, they are not very much eager to learn about the biology part of the bioinformatics. So they say that, okay, just define the job that I need to do computationally. I don't have to learn everything about the biology in the background. So I can just do the work. So it's not a good approach. It's not doing science, basically. If you're in a company as a computer scientist, then you can do the project by just with this, idea in mind, but it's not possible to do it in science. And also, just to give another point here, I think bioinformatics is not a homogeneous field. So it's interdisciplinary, multidisciplinary, and people, groups at universities are working in different aspects of bioinformatics and computational biology. For example, a group can be working on the biology part with more emphasis compared to the computational part. So they may be focused on analyzing the data at hand, and they can use the ready-to-use packages developed by other groups or even companies. So they are just users. So in that case, a person from a biology background can fit into that research group, learn that what is required very fast. But in the other case, for example, in my group, we developed those methods, develop those machine learning or deep learning based methods so that other researchers can use them. So in my group, maybe a person from computational background or a person from life sciences background but taken computational courses previously can fit better, learn better here. Fastly, we can say.
Thank you very much. We have another question in the chat box. Do you think cloud computing and other bioinformatics facilities in Turkey are suitable enough for students at university? Can you suggest some solutions? Dr. Beste Turanli?
I think I can skip this one because I said that I don't have enough experience. Probably Tunca may explain well, yes. Or Ezgi.
Dr. Ezgi Karaca?
I think, I mean, from my experience, I think it's quite good. Though I hear quite often from other researchers that the national grid is always full and they're not able to run their jobs on time. They are queuing, there are long queues, etc. So this is what I hear quite often, but my experience is not like this. So the infrastructure is quite good. Of course, more and more people are being more appealed to use them. But on the other hand, the managers of the Turkish grids are very approachable. So if you are finishing up a project, or I don't know, you are having a revision that you want to do and you explain it well to them, you might have an opportunity for at least for a short amount of time to have a priority. So what I mean is, at least you have a channel to have an option, to create an option for yourself to run things if you are really in need of it. And besides this, Turkey is now the partner of the largest compute infrastructure of Europe, Mare Nostrum 5, which is being established in Barcelona Supercomputing Center right at the moment. So we are going to be users of it as well, but under what terms is not clear yet. I think that this is still being discussed. So if we will be able to have an access to that machine as well, I don't think that we will have any problems anymore with national cloud computing.
Thank you very much. Dr. Tunca Dogan?
Yeah, I agree with Ezgi that national grid would be an option for Turkey, I mean. And other than that, we don't have many resources that are established, especially for our field, for the field of bioinformatics and computational biology. So all research groups are trying to establish their own servers. And if their university are offering some sort of, opportunity there, like some of the private universities are doing this, of course, their job is easier, especially for state universities. We don't have this situation, so we need to write some grants and buy some servers and maintain those servers, which is something else. It also affects the environment negatively, because that a cloud computing service that is maintained properly at a center or at a few centers would be more efficient in every way. But we are not there yet. Hopefully, I think we can get there in like 5-10 years.
Thank you very much. I would like to move on to our next question. Translational medicine is a field of research that aims to improve human longevity by determining the relevance to human disease with novel discoveries. How bioinformatics contributes to these fields? What kind of data is mostly used for this? And what are the most skilled requirements needed for this area? Do you have any thoughts, Dr. Beste Turanli?
Actually, it's the field that makes me so exciting, translational medicine, because now what we are experiencing today is evidence-based medicine, and we really have some sustainable solutions for the public health problems. And while we are gathering all these informations like molecular entities from DNA, RNA, or metabolites, proteins, so on, and we have also on the other side, we have information about the clinical entities, and they should be integrated to create a better resolution of our health solution, to create better health solutions, to improve the patient care, let's say. And I deeply believe that we cannot think that as bioinformatics is in the heart of the translational medicine. So that's why, you know, we cannot think that it will contribute to, it's a part of this field. I believe in that. And there's also a term that I read, like transformational bioinformatics. It's the discipline that will create, annotate, aggregate, and analyze all this rich biomedical data. So you were asking like what kind of a skill sets requirement, what kind of a skill requirements that's needed for this area. So as a bioinformatician, so you should know how to create the data or how to annotate, how to manipulate the data for the annotations, because it's a big problem, and how you can integrate them, how you can aggregate and analyze them. So there are many questions to be answered in the perspectives of bioinformatics. And I can say that probably you should be so team-oriented if you would like to feel in this field, translational medicine, because what you're going to interact with will be medical doctors, maybe patients. Somehow you can be in contact with them, but at least you're going to be sure that medical practitioners, doctors, physicians, so the biologists, all the researchers, experimental researchers, let's say, or other informaticians working on a different side of the project. So you're gonna be so team-oriented people to compile all these works if you're gonna work in this field.
Thank you very much. Dr. Ezgi Karaca, what do you think that this translational medicine effects to bioinformatics?
Like Beste, I also think that almost any subfield in bioinformatics falls within the interest area of translational medicine. So it could be bioinformatics could be a driving force for translational medicine, for example, to decide which molecule to target, or it could be also a tool to be used to analyze the data that is being generated. So at any stage, or to interpret the data, for example, in the case of computational structural biology, so to interpret the data that comes out. So in that sense, I think like bioinformatics is it can be placed at any stage in translational medicine. And indeed, as Beste said, like the most important skill or the personal quality that you should have is to like to work in teams and to adapt to different pace of different disciplines, and indeed to be able to learn how to learn from medical doctors, how to communicate to them, how to understand what their needs are, etc.
Thank you very much. Dr. Tunca Dogan, what do you think about translational medicine and bioinformatics?
Okay, it's very well put by both Beste and Ezgi, so I'm not going to repeat those parts, of course. Just I want to mention that bioinformatics has a very critical influence in this field, so that the collaborations are important. But the central part of this, I think we should coin the term of systems biology, because as we produce data and try to understand what biological mechanisms in our bodies and what goes wrong during a disease state, whether it's a simple common disease or a complex disease such as subtypes of cancer, we need to take a look to the situation from a systems perspective, so that the data that should be incorporated into this is obtained from different layers. So multi-omics approaches are quite important here in this situation. So structural biology has an effect, important effect, because we need to learn about how proteins are interacting with each other in both health and disease states, and what goes wrong, of course, during the disease. And also the interactions, physical interactions between the ligands and proteins, it's also important. And also there are some genes and they have some critical mutations that cause this disease, and their number is quite huge as well. So we need to have some sort of association between the genes and their variants with the diseases. And from the protein, gene, and also from the transcriptomics point of view, we need to integrate the data. We need to take a look from a systems perspective. We can use the tools from network analysis and graph theory. And also now deep learning is penetrating into this area as well. We can integrate the data and analyze the data using these tools and algorithms to understand better what's going on in terms of the mechanism at the mechanistic level, and then find the right sort of treatments.
Thank you very much. Due to the time limitation, we have to end our panel session. I would like to thank all of you for your contributions, for your answers. And I hope all of the participants enjoyed and get benefit from our panel. Thank you very much.
Thanks.
Thanks.
Thanks a lot.
So thank you all for this informative panel session. Now we will have a keynote talk by Yuval Itan. I hand over the platform to Dr. Caner Çağlar as our chairman for this session, who will continue from here. Over to you, Dr. Caner.
Right. Have a great day, everybody. I am Dr. Caner Çağlar, and I will be chairing this session, which will run around 30 minutes. There will be only one keynote speaker in this session, Dr. Yuval Itan. Unfortunately, due to an emergency, Dr. Itan was not able to attend yesterday's session, but he sent us a recording of his presentation, and today we will have a chance to watch his recording. Before we begin, I would like to briefly introduce Dr. Itan to you. Dr. Itan is a friend of mine from New York, actually, and I had a chance to work with him on a project related to obesity genetics. He's an assistant professor in the Department of Genetics and Genomic Sciences and a core member of the Charles Bronfman Institute for Personalized Medicine at the Icahn School of Medicine at Mount Sinai in New York City. The main focus of the Itan Lab is investigating human diseases for enhancing precision medicine by developing new computational methods to detect disease-causing mutations and genes in next-generation sequencing data of patients, and by performing, of course, cases control studies of patient cohorts to identify new genetic etiologies of human diseases as well. The Itan Lab applies and combines diverse approaches across computer science and biology, including machine learning, natural language processing, bioinformatics, statistical genomics, modelings and simulations, and population genetics. So today his talk is entitled New Machine Learning Approaches to Estimate the Functional Consequence of Mutations in Diverse Human Populations. Now is the time to listen to his insightful presentation. Hopefully it will be a great presentation for all of us.
Hi, everyone. Thank you very much for attending my talk. I'm very happy to be here. I would like to start by apologizing for not being in my talk yesterday. So I had all the intentions to do so, but unfortunately putting the wrong time on my calendar was the reason for missing it. So this is for me just another reason that I'm hopeful for next year for everything to be back to normal and seeing you all in person in conferences. So what I will talk about today is about the work that I have been doing in my lab over the last few years, which are actually separated into two different parts. So one is to develop methodologies to detect pathogenic mutations and genes in different human diseases. And the other is to apply all state-of-the-art methods and the ones that we developed in the lab for this purpose of identifying new genetic etiologies in patients with the purpose of really not only focusing on p-values, but also to look for biologically and physiologically meaningful discoveries. So this is just a standard bioinformatica pipeline of next generation sequencing analysis. I will just go quickly over it. So we have the reads, then it's mapped into the genome, the human genome, and then different quality control measurements are being done. And lastly, the part that is interesting for me is the annotated file of the genetic variants. And here I'm interested to interpret them, to see which of the variants in the patient are the ones that are actually causing disease. So this is a slide that I was actually showing for a few years, basically demonstrating the rationale for the work. You know, you have a few hundred of genes after filtration in a patient, and you want to know which one or which few are the pathogenic ones. So here as a joke, SMRF2 is the pathogenic one that people would think that it's the correct just because of the name, but actually, lastly, it was actually found that SMRF2 is indeed a pathogenic gene. But the example still stands. What we need to do is to look for the needle in the haystack to find which one is truly disease-causing, even if it's not the one that is yet known to cause the disease or known to be in the pathway. And the way to go, usually, which is very effective, is to first ask which genes are known to be causing the disease, and then to see in the list which of these genes are physiologically, biologically closest to genes that are known to cause the specific disease that the patient has. And it's not a trivial problem to do. So to try and tackle this problem, we thought about it as in navigation between two cities or between two places inside a specific map. So in the same way that we can use shortest distance algorithms to find the route between two places, we can do it between any two given human genes. So what you can see here in the red network is basically a network of all protein coding human genes with their interactivity connections, but this is based only on direct interactions. But so we are interested also to know if two genes that are not directly connected are actually close biologically and therefore relevant. So what we've done here was to do a shortest distance algorithm between any two given human genes in the human genome, protein coding human genes. And what we have is a map of relatedness between all human genes. And the way to use it is to take a list of candidate genes in a patient and then to prioritize them by their biological distance to the genes that are known to cause the disease. And if none is currently known to cause the disease, then we look for nearby diseases and prioritize by that. And you can look down here at the link for an easy to use online server for this purpose. Then we actually demonstrated it on a large scale. We took all 200 and something immunodeficiency causing genes, and then we used the human gene connectome that I've shown you before to predict in advance all possible candidates of primary immunodeficiencies based on biological distance and pathway enrichment. And that's a demonstration on a large scale, how you can pre-calculate. And then when you have a new gene, you don't need to do by yourself the connectome analysis, but you can just cross it with the pre-calculated list. And to extend it even further, we did it for all human diseases for which there are known disease-causing genes. It's called the closest disease genes server, where for each human gene, you can put it on the online page of the project, and it would tell you for this specific gene which diseases are known or predicted to be associated with a gene, or vice versa. You can start by the disease name, and it would tell you the known genes associated with the disease and also the novel predicted genes. And that is another problem that we tried to tackle in whole genome and whole exome data, which is a huge number of false positives. So for all of you that know genes like TPN or mucins or olfactory receptors, they are very highly mutated in patients, and therefore it's very difficult to rationalize if to remove them or not from the analysis, if they're truly disease-causing or not. So what we're showing here is that some genes are not pathogenic, but yet they actually contribute to a huge proportion of false positives due to their very high mutation rate, and therefore they're unlikely to be pathogenic in the general human populations. So we developed here to tackle that the gene damage index, the GDI, where what we did was to do a weighted accumulated CAD score, damage prediction for all variants in the specific gene in the general human population. And then the ones with the highest GDI, the outliers, are the ones that are much less likely to be pathogenic and can be very safely removed from the analysis. And down here on the online server, you can also choose the specific disease group to make it even more accurate. And the false negative rate here is very low, and you can really remove a lot of irrelevant genes. So it's a very quick and easy way to remove noise from your analysis. And this is how the server visually looks like. Then another problem that really came up over the years when I've done my different disease analysis was variant level damage prediction scores like CAD, Polyphent, and SIFT. And all these methods are actually using a fixed cutoff to determine whether a variant is benign or pathogenic. But here, what you can see is that for methods like CAD, if you apply the popular cutoff of 15, then if you look at the left of the two left genes and the distribution of their pathogenic mutation scores, then for the two genes on the left, all the pathogenic mutations would fall below the cutoff, meaning that here you have 100% false negative rates. You removed all pathogenic mutations from the analysis and the project is basically gone. And for the two genes on the right, they have high CAD scores for their pathogenic mutations. And here you have very low power to remove the false positives if you use CAD equals 15. So a fixed cutoff score cannot really work for all human genes because they should have different cutoffs tailored for the specific gene. And to resolve that, we developed the mutation significance cutoff, which is shortly the first method that can be used safely to remove false positive, meaning benign or neutral variants from your analysis. It's showing here that if you use the fixed cutoffs, you will have a very high false negative rate. But if you actually tailor the cutoff according to the specific mutations of each human gene, then you have very strong power to remove false positive variants. And I would like to emphasize here that if the CAD score or the other scores are going above the cutoff of the gene, it doesn't mean that it is pathogenic necessarily. But if it is below the cutoff, then it's extremely likely to be benign and therefore very safe to remove from your analysis. Then another issue of a lot of noise in NGS data are variants that are very highly common in your cohorts that you investigate, but they're completely absent in public databases like 1000 Genomes or NOMAD. And the reason is technically that all these population genetics big databases, they remove by their quality control many false positive variants, but they do not flag them. So for you, when you try to look for minor allele frequency, it would look like 0, which would look promising, but actually many of these variants that are not in the public databases are not there because they're low quality for some reason. So what we've done was to generate a list of these variants that are common in patient cohorts, but completely absent from public databases. And here we show the many different reasons of why and the characteristics of all these blacklist variants, and it can be allele elements, GC-rich regions, and other issues. But the bottom line is that when you cross your list of variants with the blacklist, and you remove all the blacklist variants, then it really removes a lot of the noise, and truly pathogenic genes can come up in your gene association analysis after that. Then to make visualization nicer for population genetic studies, we developed PopViz, which can take into consideration minor allele frequency, different human populations, and different damage prediction scores for the entity of the gene. And then according to your interest, you can also add your mutations of interest into this plot. And it will basically show you the distribution of mutations in this specific human gene and for specific damage prediction scores, which can help visually to prioritize your variants. And it's also good, I think, personally for publications if you want to demonstrate the distribution of variants in your gene of interest. Then we developed SigTailor, which is the first method, surprisingly, to very easily extract genetic sequence from VCF files and from any large-scale set of genetic variants and also of ranges of variation like a bed file. So what you can do is to upload a part of or a full VCF file, and it would provide to you either the DNA sequence or the protein sequence, which can be, of course, very useful in many different types of analysis where you need the actual genetic sequence around the genetic variant. Then this is a method that I'm very excited about, and I encourage everyone to contact me if you try to use it. I think it can have a really huge potential in the field. So it's called NHC, and it's coming to address shortcomings of methods like SCAT and other gene associations, where what you need to have a genome-wide significant signal is very high enrichment of a specific variant or a specific gene in cases versus controls. And it would not work in a few cases, especially when there is genetic heterogeneity, many different genes underlying the same disease in the cohort you're investigating. In that case, none of the genes would actually show strong signal and you would find nothing. And it would also not work in small cohorts in 100 or less, where there is just not enough power to make significance for a gene or a variant. And this method is actually thriving on heterogeneity. What it is doing is to take this philosophy of the human gene connectome of biological relatedness between genes and to look in cases for a group of genes that are physiologically, biologically close to each other, but that do not exist in high proportion in the patients, in the controls population. So just very quickly, what it's doing is to go through each gene in each patient, and it's up to you to provide a list of genes per patient after your own filtration based on the disease. So again, it's going per gene per patient, and for each gene, it is looking for the gene biologically closest to it across all genes in all other patients. And by that, it is gradually extending the cluster until scanning all genes in all patients. And then it's merging clusters that are biologically close to each other. So the end result is statistically significant clusters based on biological information that do not exist in the controls or exist to a much lesser proportion. And it's extremely efficient for small and medium-sized cohorts to really identify biologically meaningful several genes for a specific disease of interest. This is how we partly validated this method. We took a herpes simplex encephalitis cohort of 120 patients, for which 13 patients had known pathogenic mutations. And by this method, we identified in an unbiased way all the 13 genes and mutations, and also 4 novel genes which we experimentally validated, and we also repeated it without the known patients to avoid any bias, and the same top cluster showed up also here. So it seems to be working very well, but it's new, and we are very appreciative of any comments and any users trying to test it. So I'll move forward to the ongoing project. So the first one is a problem of trying to computationally identify whether a mutation is gain of function or loss of function. And we can see here that in the example of a gain and loss of function mutations, they cluster on different domains of the protein. And GOF and LOF mutations, in many cases, they result in very different phenotypes when they are in the same gene. So it's really important to know whether it's GOF and LOF. But the problem is that current methods like CAD, polyphen, and SIFT, they cannot really separate GOF from LOF mutations because they were not trained on that. They were trained on deleteriousness. So there are only two methods that were actually trying somehow to do that, to detect GOF and LOF computationally, but they don't work. One was trained only on 2 genes, and for both of them, there is no available tool to really do that. So we really want to do it on a large scale and to make it work properly. So step 1 is to construct a database of GOF and LOF mutation, a big enough one to be sufficient for machine learning. Then to very extensively annotate these mutations, and then to look for feature selection, to look for features that can separate GOF and LOF mutations. So this work was done by Chidem Sevillay, who was a postdoc in my lab, and she did a really great project and work here, starting by doing natural language processing on abstracts of pathogenic mutations, and by that she generated the biggest database to date of GOF and LOF mutations, over 1,000 GOF and over 11,000 LOF mutations. This is an example of why we actually need the NLP and not just simple text mining, because you can have many different ways to write the same term, so it needs to be done smartly. Then the GOF and LOF mutations were annotated at the gene level, protein level, and at the variant level, but by different features. And then feature selection was done to look for features that can separate GOF from LOF mutations. So let me go a bit over that. So you can see here that there are many different differences between these two categories. For example, LOF mutations tend to be frameshift mutations, and also tend to be high impact mutations, while GOF mutations tend to be in moderate impact mutations. Also, we can see that a very strong feature that can separate the classes, actually this is the strongest features by some methods, is the number of paralogs, the number of copies that the gene has. So GOF mutations tend to have many paralogs, which makes sense because this gene would have a functional backup, and then it could develop a new function. But the other way around, it would be for LOF mutations, which tend to be in singletons, where the gene does not have any functional copy, and then a mutation would result in a loss of function, lower protein expression. And we can see here that there is also a big difference in some amino acids between GOF and LOF mutations, and also about the specific domain where the mutation is. And this is just showing visually all the important features by Baruta random forest feature selection. And we repeated the process with a features exact analysis to look for protein domains and other gene and variant level features that can biologically be meaningful. And like in the other projects, we made available here user-friendly online server where you can look at the gene level and also at the variant level, and you can also choose whatever kinds of features you want to be displayed. And you can also download the full database of GOF and LOF mutations for all of your studies if you want to do the analysis locally. Now that is, so the previous work was published recently, and this is a completely new work done by David Stein, who is a PhD student at my lab, which is to create the classifier that would separate GOF from LOF mutations. Where you could put your variants of interest, and output would be whether it is neutral, gain of function, or loss of function predicted mutation. So David followed up on the previous study of CHIDEM, and he added thousands of features at the network level, from GTEx, transcriptomics, epigenetics, basically everything we could find to see which ones are most effective to separate the two classes. And you can see here that actually it makes the method extremely strong to separate GOF from LOF mutations compared to the other methods that are very limited because we're not trained on that. And this is showing how to separate LOF from GOF and neutral, which is what was previously done technically by other methods. And also here, just for detecting LOF mutations, it is also much more powerful than the other methods. So I think it has a very high potential to be very effective. And we see here visually that by LDA supervised machine learning clustering, there is a very nice separation between the three classes, neutral, GOF, and LOF variants. And the last ongoing methods project I'm going to present to you is the prediction of pathogenic mutations. So like we mentioned before, there are many methods to do that, but when we tried to rank in patients' exomes where the true pathogenic mutation in this patient is being ranked, and it's number 55 on average. So while statistically it is okay, it would show high statistical significance, nobody is looking at mutation number 55. So we want to be much more specific. And for being more specific, what we've done in this ongoing project was to classify mutations, not all of them together, like was previously done, but based on the disease they are causing. So you can see here that it's actually working really nicely. Again, by our preliminary results, we assigned each mutation to each HPO group, to the general disease group that is causing, and then using the method, all the features I've shown you before, we see also by LDA supervised clustering that it separates really nicely by disease groups, and also from neutral variants, which is in the middle, being a very tight group. So this is demonstrating that there is a really huge potential for not only detecting pathogenic mutations, but also given a new variant to say if it's neutral, or to predict which disease it is going to cause in high accuracy. And this is just the future plans to even extend more the specific classifier and the annotations. I will skip that. It's just the deep learning pipeline to do so in the same way that we are doing it for computer vision and to classify pictures of animals. We will do it for disease groups, and so far it seems to be highly effective. And here I will just briefly show you some applications of these methods, which is our actually interest, to find the pathogenic mutations and new genes causing different diseases. So this is demonstrating how it was applied. So here we have a cohort of Ashkenazi Jews that are very susceptible for having inflammatory bowel disease, IBD. And we use a whole pipeline of quality control, genetically identifying AJs, and then applying the methods I was showing to you before to identify the new genetic etiology. So firstly, we used admixture and PCA to accurately identify Ashkenazi Jews in the reference panel. Then we also used our BioMe biobank to make it even more accurate and also do some sanity tests to see that we really identify true AJs. We did power calculations to see that we have enough power, and indeed we do have currently. And then we applied the full pipeline of annotation and all the methods I've shown to you before, removing the noise and prioritizing. And what we identified was a list of genes that are genome-wide significant, which we also simulated by the human gene connectome to show that these genes are biologically closer to known IBD genes compared to random sets of human genes. So this is showing a significant physiological relevance. And then we used 4 non-overlapping pathway enrichment methods. And by doing the intersection, we came up with 7 genes that are very likely to be pathogenic and novel genes in IBD. And this is just a summary visually at the variant, gene, and the pathway levels. We also further demonstrated the likelihood of the genes to be pathogenic by RNA sequencing by polygenic risk score, where we demonstrated that combining high impact rare and also common variants can be very good to predict risk for IBD in Ashkenazi Jews. And we also did phenome-wide association studies to identify in a completely separate database of the UK Biobank that many of the enriched phenotypes for the genes we discovered are very close to IBD or IBD. And this is also demonstrating by single-cell RNA sequencing data that we had of IBD, that the genes we found are being overall underexpressed significantly in relevant cell types. So our future work, well, we have many plans for future work. I'm just showing to you 2 here. So I'm very happy to be starting a grant, an international grant of 8 different PIs around the world, together with Professor Tayfun El-Salik of the Ladakh Foundation, where we will investigate the genetic and genomic mechanisms of human brown tissues. And this would very likely be very effective for future therapeutics, and also to understand the very important basic mechanism that is very understudied until today. And we're also interested in looking for population specific disease genomics, basically looking for genes and variants that can cause disease in one population but not the other, but also looking for the overlaps to make the predictions much more accurate for individual patients. So here we plan to do very large-scale phenome-wide association studies in different biobanks across all different disease groups to identify pleiotropy, also which genes cause which disease and in which population. And this is just, again, a demonstration of a different project, COVID-19, severe cases of COVID-19, where I will just skip that. We can also show that by this specific approach and by the population specific way of analysis, we can find novel genes and also to replicate the known pathways that are currently known to be involved in the COVID-19 pathogenesis. So I would like to thank all my many collaborators and all the great people that worked with me on this project, my student postdocs, and everyone involved in this research. And if you have any questions, please feel free to email me at any time. Thank you very much for listening. Bye-bye.
So thank you, dear all. You can write your questions on chat or Discord. We are going to ask Dr. Yuval Itan on behalf of you and send the answers to you from Discord. So, dear Dr. Caner, do you want to add something?
Yeah, I mean, this is the end of the session. As you said, there won't be any Q&A part for this for the session, and you guys can send us any questions you may have, and we will email your questions to him. Thank you very much to all who attended this session, and have a great day.
So thank you. Now I would like to invite Mrs. Esra Büşra Işık, co-chair of the 5th International Symposium on Bioinformatics, to start award ceremony.
Hello, everyone. Am I audible? Yeah, okay, great. So before starting the award ceremony, I was going to send the feedback link from the chat, and I want to give you 5 minutes so you can fill the form for us, because as you know, these are really important. So I will wait for 5 minutes and we will start our ceremony at, for our time, it's 11:53. In 5 minutes, we will begin. Thank you. For the newcomers, we are waiting for you guys to fill up the feedback form that I have sent through chat. Let me send again. And we will begin the award ceremony in 2 minutes. Okay, hello all again. Welcome back for the award ceremony. We will begin now. Before that, let me share my screen. Yes. And I would like to ask local organizing committee to open up their cameras and celebrate the award winners with us. So please do. Okay. We're all here. Yes. So welcome back, everyone. And I would like to thank all of you for participating, for sending us your abstracts, your research. And it was a great pleasure for us to go through all of them, and for a scientific committee also, they really liked all of the research that was going on, and it was a really exciting and happy journey for us. And I hope so. I hope you guys are also had also enjoyed it with us. So, without further ado, I will start announcing the winners. So let me go. We will begin with the poster presentation winners. We will be announcing the top 3 winners, actually. Yes, so the 3rd place winner is Prediction of Antihepatitis C Peptides with Machine Learning Algorithms by Akash Saraswat, Arijit Maitra, and Bipin Sain. Thank you for your study, and it was evaluated by our scientific committee, and you have won the 3rd place for poster presentations. So we would like to clap. Congratulations. Let's continue with the 2nd place winners. And the 2nd place goes to Blood Group Susceptibility Shifts in COVID-19 Variant Infection Rates by Etreyi Mandal, Monisha Krishan, and Porja K. We would like to congratulate you again, and yes, congratulations. And our 1st place winner, our winner is Ligand-based pharmacophore modeling and virtual screening studies for the discovery of natural product-based PDE3 for dual inhibitors by Jia Lelim and Anand Gurao. We would like to congratulate you again. Okay, thank you all for sending us your studies, and believe us, it was really hard to evaluate the results because all of them are great, all of them are amazing, and we wish best for your future studies. So I will continue with the oral presentation winners. And the 3rd place winner is Evaluation of the impact of DNA mismatches in PCR primers to detect SARS-CoV-2 variants of concern using a thermodynamic mode by Pamela Miranda and Gerald Weber. Yes, congratulate you. And 2nd place winner is Saituna, a network alignment approach for integrating multiple single-cell RNA-seq datasets by Yasin Maruf, Hilal Kazan, and Jashim Ertan. We again congratulate you as local organizing committee. Yes. And 1st place winner for our oral presentation is, should I wait for a while? Okay, it goes to Decoding Molecular Signatures of Pituitary Neuroendocrine Tumor Subtypes Through Systems Biomedicine Perspective by Büşra Aydın, Esra Yıldırım, and Beste Turanli. We again congratulate you as local organizing committee, and thank you so much for sending us your research. And we will get in touch with you. We will contact you for certificates and all that. Again, we congratulate you. There's something in the chat. Yes. All right. Thank you. Thank you so much. So we are going back to Nihan. Let me stop my share and
So thank you, Mrs. Esra. Well, that's all. You really captured the essence of the symposium. We hope that you have had a productive 2.5 days. Now I would like to invite Dr. Mohamed Asif Khan, President of APBioNet, to deliver his closing remarks. The floor is yours.
Excuse me. Thank you, Nihan. Can you hear me?
Yes.
Okay, great. Thank you. So we have come to the end of InSyB 2021. Let me absorb that. This is a gift towards the end of the year. So I must say it has been a long and an enduring process to come to this finale. There has been a lot that has gone into making this happen, to make it, to bring it to you the way it has come up. So I want to begin by first thanking the organizing committee led by the co-chairs Aisha and Esra for all the hard work that they all had put in. This would have not been possible without them. And even though this might take some time, I actually want to read the names of all those that I can think of as my deepest, sincere appreciation for all the work. And they did all this with very minimal guidance. So they're quite independent, and they were quite energetic and actually quite entrepreneur in pushing this and making this happen. So I want to mention the names of the co-chairs: Dr. Aisha Fatima, Ms. Ashra Bushra Ishik, for leading the team, Secretary Malike Karaca, then Public Resources and Outreach team, including Gokchen Sahin, Fatma Zahra Chagil. Apologies if I pronounce your names wrongly or not correctly. Melda Mermer, Hatice Kabul, Malike Aktash, Mehmet Chebechi, Merve Sheker, Mahir Aka. And under logistics, Nihan Sultan Milat, who is here with us, Birgul Cholak Al, Ayub Salim Unlu. And we also have a few volunteers who have helped out during the actual day of the event: Rumeza Marich, Mohammed Chelik, and Farouk Ustunov. I hope I didn't miss anybody. Thank you all for all the support, all the wonderful work that you have given to this. And I know there were times when you had to meet over the weekend at night, and I know that's a big sacrifice from all of you. So again, I can't thank you enough. So I want to request everybody virtually to join me for a round of applause to the team. All right, thank you so much. Now, next, I want to thank all the participants. Of course, this could not have happened without all the participants. And you guys have been wonderful audience. You guys have been, supporting and joining and hopefully learning a lot from from the activities of the last 3 days. And although we actually had a much higher number of registrations, but this is kind of a typical for a free event. You have a lot of registration, but the actual participation is actually much smaller. But that's okay. That's normal. And I think the key is those who are really serious would come, and many of you came. And I'm happy to say that we had more than 200 actually, considering across the channels, who actually joined. This is actually the highest ever for InSyB. So this is a kind of a record milestone from the team, from the audience, and showing their support. And this is basically the bar has been set for the next InSyB to reach greater heights. So next, I want to move on to the winners of the Best Poster and Oral Presentation Award. Hearty congratulations from our side. As Esra said, there were just so many very good presentations. It was a very difficult decision, but we had to choose, and we had identified the winners, and we will be reaching out to you for your certificate. So congratulations once again. And I just want to highlight that as the president of APBioNet, the Asia Pacific Bioinformatics Network, Turkey is kind of in a unique position because part of it is in Asia, part of it is in Europe, and it bridges the two continents. So this is a very special InSyB for us, being in a location that bridges two continents. And this is also our first time coming to Turkey. So this is a very historic event. And I hope this is not just the first, and this is not the last, but actually the first of many more to come. And on behalf of APBioNet, I want to thank Beykoz Institute of Life Sciences and Biotechnology, DILSAB, and of course, Bezmi Alem Mokkaf University, the parent organization of DILSAB, for organizing this event, and for the support we got from the International Society for Computational Biology, ISCB Regional Student Group, RSG Turkey, and it was very kind of the president of TÜBİTAK, the Scientific and Technological Research Council of Turkey, to grace our event. So a big thank you to the organizers, organizing entities, and also the support we got from RSG, ICB Turkey, and TÜBİTAK. So last but not least, I hope all of you enjoyed from this experience, learned a lot from this, and excuse me, and will not kind of stop there, but actually would take this as a step forward for your journey in bioinformatics. The videos are available, so if you missed anything, you want to recap something from what was covered, you would be able to come back to our YouTube channel. And I also hope you will use this opportunity to stay active in the Discord channel, make connections, and who knows what these little sparks of connections would do to your life in a bright, positive way. So all the best, and also wishing all of you a happy new year in advance. And we look forward to much more exciting activities next year from APBioNet, from BILSA, Bezmi Ali Markov University, RSG, to BITAC, to spread the love for bioinformatics in the country and beyond. Thank you very much, and back to you, Nihan.
So thank you. I would like to once again to thank all our participants, our speakers, all of you. So we will end our session here. Thank you, everyone, and have a great day ahead. See you in our next events. And now we can proceed with Esra.
Hi all. I just wanted to thank everyone by, again, a presentation. Sorry for the misunderstanding. So yes, we have prepared we had been preparing since September as a team, and as Dr. Asif had read their names and all that, but I want to go through all of our sports team actually. First, I want to thank Bezmi Ali Markov University for allowing us and helping us for every step of this symposium, and for hosting this symposium. And I want to thank APBioNet for creating such an event so that we can connect with people, we can share everything with other researchers in this field. And I want to thank RSG, ISCB, RSG for supporting us and for creating an amazing panel session with amazing speakers. And last of all, I want to thank F1000 Research, which we will be publishing our abstracts. And I want to remind you that if you want to change anything, if you want to add anything, please share it with us as soon as possible, mail us, because we will be getting in touch with F1000, and the final versions of your abstracts will be sent. And I want to thank our advisory committee, our rector, Professor Dr. Rümeyza Kazancıoğlu, our director, Professor Dr. Mehmet Ziya Doymaz, and our supervisor, Dr. Muhammed Asif Khan, for advising us, for being with us during this session, during this journey, and also for being one of our opening speakers. And I want to thank our scientific program committee, because they had been very much active for all of the abstracts, for all of the presentations. They didn't say, they were always supporting us as a student team. We were always asking them what should we do, and they were always guiding us. And as I said, they have tried so hard to be fair on this journey. And I will not be able to read all of their names, but especially Professor Atilla Akdemir, who was the chair of the scientific program committee. He was also really helpful during this journey. I want to thank all of them from the bottom of my heart. And I want to thank our keynote speakers. They are not here, but in case they watch the YouTube video later. So I want to thank all of them for accepting to be a keynote speaker for our InSyB 2021, because this was an international event, and as you're also aware, the time differences can create many problems. We had speakers from US, we had speakers from Malaysia. So they didn't care about the time differences. They just wanted to help us with this symposium. So I want to thank them also from bottom of my heart. And here is our local organizing committee. Dr. Asif read all of their names one by one, but I just want to show them to you. First, my co-chair, Dr. Ayşe Fatima, I really want to thank her, because we have worked together and she has been a great co-chair for me. So I want to thank also Melike. She was the secretary, and she was the one actually who was answering all of your questions in the mails. And I want to thank Melda, Gökçen, and Fatma Zehra. the PR team, they have worked really hard for advertisement of the symposium. And I want to thank Rumeysa, who had been moderating these Zoom sessions, and she, even though she was she joined us newly, she kept up with us so quickly and helped us during the sessions. And I want to thank Eyüp Selim, Birgül, and Nihan. They were the logistics team. They were working really hard for your abstracts, really hard, like to get your abstracts, to correct what was wrong. So actually, they were the key players for the presentations. So I really want to thank them, and they are here. Thank you. And I want to thank Mohammed and Faruk. They helped us as latecomers also. They really helped us with the presentations, with the orders. So they were also key persons for our sessions, for breakout rooms and everything. So without further ado, I also want to thank you, because it wouldn't be possible if you did not join, if you did not help, if you did not send us our research. So I really want to thank you, and I hope we can meet again in better or broader symposiums, conferences, or other occasions. We hope to meet you again, and we hope you all stay healthy during this time, and we wish you the best for your studies and research and your life, and take care of yourselves. Ya sen kapat. We will be closing the session now, everyone. Thank you again and take care.